Today, about 74% of drugs are taken orally and are found not to be as effective as desired. To improve such characters, Transdermal drug delivery system has emerged in today's technology. Delivering medicine to the general circulation through the skin is seen as a desirable alternative to taking it by mouth. Patients often forget to take the medicine, and even the most faithful compliant get tired of swallowing pills, especially if they must take it for several days. In several cases, oral drug administration has a huge potential to be affected by the gastrointestinal irritation and hepatic first-pass metabolism. Therefore, the innovation on Transdermal drug delivery system offer better option on delivering drugs. One of it is by the Transdermal patches. Patches have huge advantages in avoiding the hepatic first-pass metabolism, enhancement the therapeutic efficiency, and help in maintaining the plasma level. The current design of delivery patches is generally divided into two subcategories, which is the first one is the matrix types and the other one is the reservoir type. The matrix design is a simple where the medicine is embedded in the polymer matrix and easier to fabricate with controllable process parameter. However, the rate controlling membranes depends on the fabrication process and has limited flexibility compared to the reservoir type system. While the reservoir design holds the bioactive agent in a solution or a gel condition that goes about as a bioactive agent delivery, and the release of agent is frequently constrained by the rate controlling system present between the reservoir and the tissue, where the initial burst release are observed in several cases. However, it will be required high complexity in design and to compared to the matrix type system, TDS system. In our invention, the bilayer membrane was developed by electrospinning of the PVDF and PVA polymer solution, forming the first nanofiber layers. Then the electrospun we place on top of the spin coater holder, where the PVA collagen HNT hydrogel solution is dispersed on top of the electrospun layer for 30-second spinning duration before subjected to freeze thawing process for 3 cycles. The overall process forming a bilayer membrane consists of microfibrous and nanofibrous layer, which further loaded with bromidine, chlorine, and benzalkonium chloride for kinetic drug release analysis. France diffusion cell was used to stimulate the transdermal drug delivery process by using Drug-M, which is a synthetic skin membrane, and also a rat skin. So the results suggested that the bilayer have a strong and flexible mechanical properties with excellent swelling percentage and porosity properties. Scanning electron micrograph of bilayer cross-section demonstrated that the fabrication successfully intact the hydrogel surface on top of the electrospun layer. The surface roughness of the bilayer is adequate to allow the specific interaction between drugs and cell host with the matrix scaffold. The in vitro kinetic drug release analysis using France diffusion cell suggested that the bilayer membrane successfully reduced the burst release effect and solved the delay on the electrospun drug delivery process. After 310 minutes, the bilayer TDD membrane system continuously released the drugs, which show the potential of this project to be used for long-term treatment. The main novelty or invention in this project is on the drug-in-adhesive material designs that consists of PVDF and PVA nanofiber electrospun, and also PVA collagen HNT macrospore structure from the hydrogel layer, which producing stable and superior drug reservoir bilayer membrane. This combination of two layers will help to reduce the burst release effect and prolong the drug release at the targeted area for better healing process. Besides, the combination of the structure gives huge flexibility on the drug solution due to simple and economical fabrication process. The morphological structure of the matrix that mimicking the extracellular matrix will help to channel the formation of new tissues during the healing process that help to promote or to eliminate the scar formation at the lesion site.
The benefit to society is first, new approach on modification of biomedical scaffold as template for transdermal drug delivery with controlled drug release. It helps to reduce burst release of drug in early stage due to their nanofiber Electrospun-Hydrogel combination, which can enhance the application of transdermal drug delivery. Second, new alternative for transdermal patch product with product sterility condition that can reduce time during treatment. The simple fabrication process can create a drug-in-adhesive patch at controlled dimension with varying chemistry and topography. And third, increased rate of mortality by help ensuring patients keep getting consistent dosage of drug during the treatment. The controllable resist properties help to minimize human error, especially for old folks and memory-related patients. The process used allowed to be tailor-made according to patient dimension, and the bilayer membrane can be used with wide range of drug concentration. The materials used in the fabrication process does not produce unwanted byproduct which can cause harmful effect to the living organism and the environment. Part of the material is derived from industrial waste such as collagen from fish scale and bromelain from pineapple stem. The electrospinning process does not require use of coagulation chemistry or high temperature to produce solid thread from the mixture solution. Therefore, excessive amount of chemical and energy issue will not arise. As mentioned before, the in vitro cytotoxicity study confirmed the mixture of material informing the matrix membrane not causing inflammatory response toward host tissue or cells. Transdermal patches market is expected to gain market growth in the forecast period of 2021 to 2028. Data Bridge Market Research analyzes the market to grow at a CAGR of 5.75% in the above-mentioned forecast period. Increasing prevalence of chronic disease drive the transdermal patches market. Transdermal patches are the type of adhesive patches which are medicated. They help in delivering a present dose of medication inside the body by using transdermal technology and diffusion technique. The medication later gets into bloodstream over period. These patches are used for various diseases including migraines, diabetes, asthma, cardiac and hormonal disorder, among others. As this project can be implemented into various number of applications, we can have a lot of potential customers, especially related to healthcare, such as WHO, Asians, KKM, hospital, and other private institutions. With this technology, we also can tailor the patch into therapeutic segment of IKS. For example, as cosmetic product where this invention it could help to boost up our economy system by serving as local manufacturer. Our nearest competitors are from international brands that focusing more on specific medical problem where they tailor their TDD patches for specific drug. However, our invention can tailor-made according to type of drug, either it is synthetic or bioactive compound itself. We have collaborated with Abdul Muaz Food Enterprise in terms of potential of bromelain in therapeutic application and Legacy Megajaya Sendirian Berhad in the TDD patch material development. We also have patent a part of the fabrication process, especially for wound care TDD system application. Hope in future, few items will be documented as copyright or patent. Our project has also successfully published in several indices, journals, proceedings, and book chapter. At the same time, we already submitted few recent findings to publisher to publish the manuscript. Thank you for lending your ears and your time. I hope that our present work catches your attention. Stay safe, kita jaga kita, stop spreading COVID-19.